today we are going to start with the primary and secondary aspects of motivation. The motivational potential to perform various types of activity is sometimes called the primary aspect of motivation. The primary aspect of motivation is nothing else but the motivational potential to perform various types of activity. But the secondary aspect is the characteristic of behavior that the animal exhibits at a particular time, like the primary thirst is brought about by the dehydration of body tissues and is represented in the brain as the motivational potential for drinking. In addition to being affected by primary thirst, the primary thirst is the reflecting basis bodily needs. The primary thirst is the reflecting basic bodily needs. And this primary thirst of drinking is affected by secondary thirst, contributed by the palatability of the liquid, drinking in association with meals, etc. Although the necessity for distinction between primary and secondary aspects of motivation can be recognized by direct observation of behavior, the details of the mechanisms are best explained by experiments. So, the primary motivational states are investigated in the animals kept in strict controlled regime, maintaining day length, temperature, etc. But suppose we have to investigate the effect of ambient temperature upon drinking in pigeons. We are going to investigate the effect of ambient temperature upon drinking in pigeons. The experimenter has to maintain pigeons for 2 days without water at a particular temperature. Then the standard tests are done and it is seen how much the pigeons drink. The experiments are repeated with different temperature maintenance. The standard tests are always conducted by providing standard drinking bowl, standard cage at a standard temperature. If differences are noticed in the amount of water drunk during each test, these must be due to differences in maintenance temperature, since it is the only factor allowed to vary in each experiment. So what are we doing here? We are going to investigate the effect of ambient temperature upon drinking in pigeons. So we are going to keep the standard conditions by maintaining standard cages for them, standard drinking bowl, and all those standard things. But we are going to change the standard temperature. That is the only thing which we can alter. So the variation in temperature can only induce changes in primary motivation. The primary motivation is the animal's potential for drinking under steady state conditions. Now we are going to learn how to study the secondary aspects of thirst. As we have mentioned earlier, the secondary thirst is contributed by the palatability of the liquid, drinking in association with meals, etc. So we are going to learn about how to study the secondary aspects. The maintenance of conditions are held constant and the selected testing condition is varied. The bird is deprived of water for 2 days after at the same temperature. Here we are measuring the secondary aspects of thirst. So we are going to keep the bird away from water as water deprived for 2 days, always at the same temperature. Here we are keeping the temperature as a standard temperature without changing the temperature, always at the same level. And standard tests are carried out in standard cages with a standard bowl, etc. Only the ambient temperature is varied from one occasion to the next. Any differences noticed in the amount of water drunk during these tests must be due to temperature differences at the time of drinking. No difference was observed in primary motivation from one experiment to the other because the conditions under which the animals are deprived of water are always the same. Therefore, any differences in the amount drunk must be due to changes in secondary motivation, and as we have told earlier, the secondary motivation is something which is related to the ambient temperature variation. Okay. In other experiment, it is seen that the higher the ambient temperature, the more is the water drunk. When we increase the standard temperature, more water is being drunk by the pigeon. This suggests that water drink in response to a given ambient temperature in anticipation of the water that they may need for cooling themselves rather than drinking to make up the water that they have already lost in cooling. In general, the primary motivational states are concerned with relation between animals' physiological state and its behavior. The maintenance of a stable internal physiological condition is an important aspect of motivation. Thus, hunger and eating are to maintain a satisfactory energy balance, and the water balance of the body is maintained partly through the mechanism of thirst and drinking. The studies of secondary motivational states are concerned with the more psychological aspects of motivation. For example, highly palatable food increases the feeding incentive of hungry animals. If the consequences of this are less than the animal has been led to expect, the motivational state of the animal will be considerably altered as a result of frustration. The studies of secondary motivation states are concerned with the more physiological aspects of motivation. For example, highly palatable food increase the feeding incentive of hungry animals. If the consequence of this are less than the animal has been led to expect, the motivational state of the animal will be considerably altered as a result of frustration. A hungry animal is likely to become frustrated if it is physically prevented from obtaining food, which it can see if an expected food reward is delayed, or it is if its food is less palatable than usual. Now we are moving on to the next topic, that is nothing else but the deprivation and motivation. The another important aspect of motivational studies is to note whether or not depriving animals of opportunity to perform a particular activity causes a build-up of motivation and results in an increase in behavior when the deprivation is over. Deprivation of food will result in increased feeding. That is happening because of the build-up of hunger, and of sleep will result in increased sleep due to the accumulated tiredness. But avoidance of a stimuli motivated by fear is not enhanced by deprivation of those stimuli. So the deprivation causes a build-up of motivation in two cases, such as the increased feeding case and increased sleep case. But according to the motivated behavior of fear, it is not enhanced by building up. Okay. It is also important to notice that there is a build-up of aggressive motivation when opportunities for aggressive behavior are denied. To cut this, here we can see an example. Males of yellow damselfish behave aggressively towards the males of their own species if presented to them behind a glass partition. Here we are going to use a fish as a tool. Yellow damselfish behave aggressively towards the males of their own species if presented to them behind a glass partition. After being deprived of the opportunity for 7 days, a fivefold increase in aggressive behavior was observed. There had been a build-up of aggressive motivation during the deprived period. Now we are going to learn about the build-up of sexual motivation. that can be exemplified by the experiments on zebra finches inhabiting semi-arid plains of Australia and showing highly social ways of life. The separation of male and female have shown that the male is strongly motivated towards reunion with the female and will actively search for her. The longer the separation, the more is the upsurge of sexual activity between the pair. There are generally three main features of male behavior which can be taken as typical motivational causes, as the male appear to show a buildup of sexual desire during periods of separation from mates. Males separated from their mates exhibit typical appetitive behavior in the sense calling behavior or searching for the mate, etc. And the isolated males will learn an apparent response rewarded when the sight of another finch mate. They show an apparent response rewarded by the sight of another finch. Now, let's move on to the next topic. That is about the physiological basis of motivation. Besides investigating the motivational behavior or drive with the help of examples, it is also important to examine motivation at physiological level. On the neurophysiological side, motivated behavior is actually an instinctive act, and the motivated behavior has been investigated by tracing out parts of the nervous system involved and the effects produced on them by the external stimuli and changes in the internal environment. The area of vertebrate brain which has been considered important in control of motivation is the hypothalamus, which is situated in the floor of the diencephalon below the thalamus. It is in the thalami. Thalami means the side walls of diencephalon. It is the hypothalamus. It is in thalami fiber tracts link up with one another in the numerous nuclei or clusters of neuron bodies. Hypothalamus has its intimate connection with pituitary. Pituitary gland is also called as the master endocrine gland. So, which parts are associated here? Hypothalamus and the pituitary. The hypothalamus has been found to contain excitatory mechanisms contributing arousal of motivation and inhibitory mechanisms reducing motivation. These two extremes can be seen, and the former are controlled from the lateral sides, whereas the latter form the ventromedial area of hypothalamus. The excitatory mechanism is actually controlled from the lateral sides, whereas the latter, that is the inhibitory mechanism, is controlled by the ventromedial area of hypothalamus. Two regions are very important. The lateral sides of hypothalamus, which controls the excitatory mechanism and motivation, and the ventromedial area of hypothalamus, which contributes to the inhibitory mechanisms. As for example, electrical stimulation of the lateral hypothalamus produces increased eating, and that of ventromedial hypothalamus causes a reduction in eating. When we conduct experiments by manipulating the hypothalamus, in case of rats, we can see two examples. When we electrically stimulate the lateral hypothalamus, it produces an increased eating in these rats. But when we electrically stimulate the ventromedial hypothalamus, it causes a reduction in eating. Hypothalamus has been found to monitor the thirst, sexual behavior, emotional behavior, sleep and maternal behavior, and these findings have led to the conclusion that drive or motivation is based on excitatory mechanism and satiation or satisfaction is based on inhibitory mechanisms of hypothalamus. When we learn about motivation in ethology, we have learned that there are two important terms which we, you know, usually hear, which are the drive and the satiation or satisfaction. So, this drive of the motivation is actually associated with the excitatory mechanism of hypothalamus in case of food or sleep or something else, and satiation or satisfaction is actually related to the inhibitory mechanisms of hypothalamus. It is also believed that action of both the aforesaid mechanisms is controlled by relevant sensory stimulation, changes in internal environment, and influences from the cerebral cortex. Now let's learn about the role of hypothalamus in thirst and hunger. This is something which we learn to connect this particular ethology subject with our daily life by connecting this with the major topics of thirst and hunger. The thirst and hunger drives are quite necessary in maintaining a balanced psychological and physiological state, or the homeostatic state. If a mammal loses water from its body, the blood becomes hypertonic. Hypertonic. When a mammal loses water from its body, the blood becomes hypertonic. This change in concentration is detected by receptors or osmoreceptors present there in the lateral hypothalamus. Two compensating systems are set in by these receptor cells via a link with pituitary system, causing secretion of ADH or antidiuretic hormone and resulting reabsorption of water by the kidneys, causing the animal to seek out water to drink. If the link between hypothalamus and posterior lobe of pituitary is damaged, ADH or antidiuretic hormone may never be secreted, and a condition, diabetes insipidus, that is the copious urine discharge, is developed. In an experiment, a goat was injected with a few thousands of cc of hypertonic saline solution into the hypothalamus. The goat stops whatever it was doing, runs over to the water through and drinks as much as 7 liters of water. So the sensation of thirst is actually related to the saline solution, or the salinity of presence of saline solution in the hypothalamus. So this makes the blood become hypertonic. That leads to activation of osmoreceptors. Many systems are activated, like the pituitary system, and which is related to something called as the ADH. And finally, all these processes leads to the animal to seek out water to drink. In an experiment, we have mentioned a goat was injected with a few thousand cc of hypertonic saline solution into the hypothalamus, and it caused that animal to drink excessive amount of water. Rats with damaged ventromedial part of hypothalamus lose sensitive control of their feeding. They eat almost constantly and become so fatty that they cannot eat even more, or they can't even move. Electrical stimulation of this area depresses feeding, so that the ventromedial area has been called a satiety center. The complementary center, which promotes feeding, is situated in lateral hypothalamus, closely associated with drinking area. This feeding center initiates the appetitive behavior of eating. If this area is electrically stimulated, feeling is elicited, and if damage is done, it can lead to starvation in the presence of food. Now let's learn about the limbic system and its behavior. While hypothalamus is essential for initiation of limbic system and behavior, While hypothalamus is essential for initiation of motivational states, other parts of the brain are also involved in their control. Parts of the forebrain are also found involved in feeding, drinking, sexual, and aggressive behavior. Lesions in the frontal lobes of the cerebral hemisphere and limbic system, which is situated near the base of cerebral hemisphere linked with hypothalamus, involved in general aspects of behavior, modifying the activity of hypothalamus in mammals. All these portions affect the sexual and feeding tendencies of mammals. Dogs with damage in parts of frontal lobe show greedy eating, devour inedible materials, and become fatty. In case of cats, the lesions in the region of amygdala, a part of the limbic system, causes hypersexuality, attempting to mate with other animals and even with inanimate objects. So, amygdala in case of cats is a part which is related with sexuality of these animals. When lesions are caused in this region in cats, this causes hypersexuality. The anatomists who worked out the interconnections of the limbic system proposed that it was concerned with emotional responses. It has been found that destruction of neocortex produces extreme placidity or peacefulness in cats. Destruction of neocortex produces extreme placidity or peacefulness in cats, suggesting that the neocortex is essential for aggressive motivational state. Electrical stimulation of the same parts of the brain cause the animal to attack any available person or object. Ferocity or aggressiveness. All these characters are caused by electrically stimulating the neocortex region. This cortical mechanisms have their influence to hypothalamus, not to be important in the arousal and control of aggressive behavior. Arousal and control of aggressive behavior is something which is related with the hypothalamus. When we electrically stimulate the neocortex in cats, that produces aggressive behavior. Self-stimulation of brain and motivation. The role of hypothalamus and other Ryan cephalic in the sense for brain related structures and motivation is explained through somewhat different kinds of experiments. In this case, an animal such as rat, cat, or monkey is chronically implanted with an electrode in the lateral hypothalamus. The electrode was connected to a stimulator by a thin wire. The animal was placed in the situation where every time it pushes on a lever, a brief current is then flowed, stimulating the brain of the 1/2 of a second in an experiment with rat. The latter pushed the lever first as fast as possible, up to 5,000 times per hour. The rate of lever pressing increases when the animal is deprived of food and falls markedly just after the animal is fed. That's it. Self-stimulation behavior is strongly motivated, as shown by the fact that the animals will push the lever many times for just a single stimulation, and they will walk across the electrically stimulated grid to push the lever more readily than they will to eat when they are very hungry. So that is something which is related with the self-stimulation of brain and motivation. Self-stimulation behavior is strongly motivated, and it is shown by the fact that animals will push the lever to get that electrical stimulation many times for just a single stimulation, and they will walk across the electrified grid to push the lever more readily than they will to eat when they are very hungry. As a conclusion, in higher animals, phylogenetically, the motivational aspects of instinctive behavior emerge clearly after the animal have begun to learn, and they are generally talked about in terms of drives, goal-oriented behavior, and satiation. In mammals, it is believed that the arousal of drive is the result of activation of an excitatory neural mechanism in the hypothalamus and the reduction of drive or satiation, the activation of inhibitory hypothalamic mechanism. It is thought these basic mechanisms that the combined effects of the internal environment, sensory stimuli, cortical influences, and learning regulate motivation. And some scientists have shown that there is considerable plasticity in the cells of lateral hypothalamus. When lesions are created in this area, the rat stopped feeding, but it reverted back to normal feeding again. This means that the neurons outside the original feeding center have taken over this function. Now, some other scientists have doubted the specificity of the centers, and they found that different behavior feeding or drinking can be elicited from the same electrode in lateral hypothalamus depending upon the experience of the animal. It is therefore evident that stimulation at the same site on different occasions may not always produce the same results, or it should not be expected that the artificial stimulation nor the natural external stimulation will arouse only one isolated motivation system. It is therefore evident that motivation at the same site on different occasions may not always produce the same results, or it should not be expected that the artificial stimulation nor the natural environmental or external stimulation will arouse only one isolated motivational system.